Hello everyone, welcome to my channel Biozy, and in this video we are going to discuss mutations that take place in Lac Operon. So Lac Operon is basically a polycistronic structure where there are several structural genes under the control of promoter and the operator region. So we are going to discuss what happens when there is going to be mutations in the Lac Operon. So before starting with the mutation, first you should know the basic concept of the Lac Operon. So Lac Operon, if in the absence of inducer is the lactose itself here, right? So in the absence of inducer, what happens if there is no inducer? The I gene, it codes for the mRNA repressor that translate into the repressor protein. So in the absence of the lactose or the inducer, this repressor, it goes and binds to the operator region and thus prevents further binding of the RNA polymerase to the promoter region. And if there is no polymerase binding to the promoter region, there will be no transcription or translation of the structural gene, that is Z, Y and A, required for the lactose metabolism. So if there is no lactose, we don't require lactose metabolism, and thus this happens. But when there is lactose present in the medium, then what happens? If there is lactose or the inducer present in the medium, then that lactose is going to go and bind with this repressor molecule and bring about a conformational change in this repressor molecule. So when this repressor will no longer be able to bind to the operator region, this operator region is thus free, and this will further allow for the DNA dependent RNA polymerase to bind to the promoter region, and it slides and read all the codes in the structural gene, that is in ZYA, and they are going to transcribe and then translate into their respective product, that is beta galactosidase, permease and transacetylase. So this happens in the Lac Operon when in the presence or absence of the inducer. But if there is mutation is basically sudden change in the gene sequence of the DNA. So if there is change in the various segments of the Lac Operon, what is going to happen then? So in this we are going to discuss first the first case. The first case is where mutation is occurring in the I gene. So I gene, as we have seen, is the one that is coding for the repressor part. So if there is changes in this I gene, it will no longer be coding for the repressor region. So if there is mutation in I gene, no repressor that is going to form, and if there is no repressor, this repressor will not be able to bind, of course, to the operator region, and if it is not going to bind to the operator region, then normally what is going to happen, the transcription or the translation of the structural gene normally takes place. So if there is no repressor, then it leads to translation of all the structural gene Z, Y and A genes easily. Okay, now the second case is when there is mutation in the structural gene. If there is mutation in the structural gene was Z, Y and A, and they are required for the metabolism of the lactose. Like Z is coding for the beta galactosidase enzyme that breaks down the lactose into glucose and galactose into its monomeric unit. Y is required for the permease enzyme, and A is required for the transacetylase enzyme. Now if there is mutation, let's say in the Z region, Z structural gene, then in that case it will not be able to translate into the beta galactosidase enzyme. So if there is no beta galactosidase enzyme, then of course the lactose will not be broken down further into its monomeric unit. So if there is mutation in any of the structural gene, that is Z, Y or A gene, then there will be no mRNA translating into the respective product. So no product will be formed. Now the third case could be the nonsense mutation. Now what happens in the nonsense mutation? Nonsense mutation when let's say in a gene, if this is a gene and this gene is coding for one of the amino acid, but now because of nonsense mutation, this sense codon in that particular region gets changed into chain terminating codon. That is, it is going to change in one of the UAG, UGA or UAA, which are the chain terminating codon. So if in any region the chain terminating codon is happening, so it will no longer be forming the or translating into mRNA. So the transcription or the translation process stops there itself. So what is going to happen in this case? Let's say the nonsense mutation, if it is happening in the first case, let's say if it is happening in the lac Z, lac Y or lac A gene, then of course the mRNA of the gene in which mutation has occurred will not be translated. So there will be no formation of the protein for that particular gene. And let's say if this nonsense mutation occurs in the operator region. So if you remember, this operator region is the part where the repressor molecule binds, right? So if in this region, in the operator region, the changes occur, so repressor will not be able to recognize that operator and will not be able to bind it. So no repressor molecule binding to the operator region, and in that case what is going to happen? There will be no binding of repressor to the operator part. Thus it leads to translation of Z, Y and A genes. See, if there is no repressor binding to the operator region, then in that case all the structural genes will be transcribing and translating into their respective product. Thank you for joining. Keep watching. Bye.